Welcome to Optica's online industry meeting today. We'll be starting in just a minute. We're so glad you're here. We ask that you could please
Thank
keep your
you
camera
very
on.
much, Elise. We are really glad you're here. Happy New Year, everyone. It is the first online industry meeting for Optica in 2023 and the first of many. And today we have something very special for all you. Thank you for joining our online industry meeting on medical devices. Let me share two things today. First, a big thank you for all the support you have given Optica over the last year. Together, we are building a larger global industry network to promote and defend the interest of next generation photonics. Second, are you active in medical markets? Do you manufacture photonic devices for remote health monitoring? I'm talking about wearables, DNA sequencers, or breath analyzers. If so, mark January 17th in your agenda. It was back in August 2021 that Rockley Photonics rang the bell on the New York Stock Exchange.
So we're making chips that are processing, sensing, and communicating information optically as opposed to electronically.
By building patented silicon photonics-based laser technology into a new generation of small modular sensors, they claim to significantly expand the range of biomarkers that can be detected and measured non-invasively by wearable devices. So, what has happened in the meantime? And more important, how can we, the Photonics family, accelerate the rollout of their unique sensing platform? Is there a demand for semiconductor lasers, micro-optics, a specialty fiber for endoscopy, photonic integrated circuits? If you ask me, I would say yes. But let's ask some of the main stakeholders and get their insights. On Tuesday, January 17th, our online industry meeting is on photonics for medical devices. And yes, Rockley Photonics will be there, represented by Kate Bechtel. What do they need for their next stage of development? And joining them, we will welcome PacBio. They develop and manufacture systems for rapid gene sequencing, essential for fundamental biological research. PacBio describes its platform as single molecule real-time sequencing, SMRT, based on the properties of zero-mode waveguides. Optica friend Gloria Huffler will tell us how we can help her. Meanwhile, in Europe, glucose monitoring for diabetes is moving towards no-needle measurement thanks to Diamond Tech. This German startup has attracted more than $20 million in funding to pioneer non-invasive blood glucose measurements using their patented photothermal detection technology. On January 17th, we'll get an update on their progress. They operate in the mid-infrared. So how does the quantum cascade laser roadmap and their mid-infrared technologies match their ambitions? And when it comes to breath analyzers, we have been shocked by the fast developments of the German company Halitos. Joanna Paiva and medical doctor Sven Jungmann will demonstrate why laser spectroscopy is key to teaching machines to smell your breath for diagnostic clues, and they want to make their technology handheld. Of course, we need to talk about optical coherence tomography and ophthalmic applications. Adam Wax from Lumerica Vision will illustrate the use of OCT to screen for early signs of eye disease.
This is a device we built in my lab at Duke University. We have a patent on it. It's portable, weighing in at 4 pounds, highly affordable. The cost of goods is $5,000 on that.
So if you want to help us unveil emerging business opportunities in leading-edge medical photonics, join us on Tuesday, January 17th at 10 a.m. in Washington and 4 p.m. in Berlin for the next Optica online industry meeting. Let's make the world a healthier place. And it all starts with an early diagnosis. Thank you very much for being with us today. It is indeed 10 a.m., 10:04, and now we are ready to start. All of you are here to find cooperations. All of you are here to find partners. Over the next 2 hours, we're going to start on time, we finish on time. The goal is for all of you to find other people that can help you accelerate your process. My name is Jose Poza, and I'm here talking on behalf of Optica. Optica, the global Society for Advancing Optics and Photonics worldwide. My job is to make sure that the association, the industry network that Optica is, make sure that their corporate members find value in being part of our network. Most of you are corporate members. Thank you very much for being with us, and thank you very much for the constant support we got from you in 2022. What an impressive year. Remember that because you are corporate members, you have access to market reports, you have access to top-tier connections. We introduce our companies constantly. You also have access to a long list of events, and most important, you have access to our staff who dedicates their life to understand your technology, your applications, and try to make sure that we help you advance your business. We have a corporate engagement council. These are the people who direct us. They are the people who tell us in which direction to go. I would like to always acknowledge their help and their constant feedback. Thank you very much for your great support. This is our calendar for online industry meetings in 2023. Today we have medical devices. Write down, because at the end of February, always the last Tuesday of the month, we have a different topic. The next one is fiber sensing, and I can anticipate some endoscopy applications of that meeting. In addition, we also provide industry tutorials, product releases, and real life meetings. For those of you who are coming to Photonics West, contact us separately because we are organizing something really big in San Francisco at the same time. Remember also, because you're corporate members, you have our own list of market reports that are accessible to you. Here I highlight the one that we purchased late last year on meta materials. Please make sure that you're looking for meta lenses, or you're looking for technology to make ultra thin flat optics with capabilities similar to their 20 times thicker counterparts. This is a market report that you should read. And also, I would like to let you know that today is all about medical devices. And we have a really, really great agenda with us. We have a long list of companies presenting: Lumedica, Diamondtech, Rockley, Halitus, and PacBio. But this is not a meeting for the speakers. This is not a webinar. This is a meeting. And a meeting means that people have registered to participate at that meeting. And we have taken the time to analyze every single company that have registered to this meeting and is here, either in the Zoom room or watching from YouTube, and is looking for partnership. These are all the companies who register for this meeting. We are going to provide all the people who register with the full participant list: name, surname, company, position, and LinkedIn address. And if you want to get in touch with any of the participants today, all you have to do is send us an email and we'll make the introduction. This is, in our opinion, the most important corporate membership benefit. But we would like to start the meeting now by letting you know that in this meeting, we want everybody to be engaged and want everyone to find out what can they do to help. So every speaker will get the first question the same, which is, what can you do for the others? And what can the others do for you? So let's start. And the first presentation is one that you all have been looking for. I've been waiting to hear this presentation for a long time. Rockley Photonics. Thank you very much, Kate, for being with us today. And tell us, most important, how can we help Rockley Photonics, and how can Rockley Photonics help the entire photonic industry? The floor and the attention of everyone, about 400 people registered for this meeting, is yours, Kate.
Thank you. I don't know if I can match your energy.
Oh, you can try. It's coffee.
With how few
It's nothing
cups
less
of
than
coffee I've
coffee.
had this morning. I'm only 1 cup of coffee in, so we'll try. So let me go ahead and share my screen. And I hope you all can see that.
Crystal clear.
Great. Good morning. It's an honor to start off the session today. I've been in biophotonics for nearly 20 years now. I did my postdoc at the MIT Spectroscopy Laboratory with Michael Feld back in the day. Most of my working career was at a contract R&D co-development firm called Triple Ring Technologies, where I worked on dozens of different projects over the years, covering lots of different aspects of sensing and imaging. So some of what I'm going to talk about today is coming from that perspective of seeing a lot of the same challenges over and over again. I started at Rockley Photonics about 1.5 years ago because I was intrigued by what the silicon photonics platform is capable of. And it's capable of quite a lot. Rockley's platform is vertically integrated. It encompasses not just the PIC or photonic integrated circuit, but also the ASIC, which is the custom electronic IC that drives the system and packages the data, the firmware, the software, the mobile app, and cloud, along with algorithm development using machine learning. Our current product is the Bioptics band that is shown here. And there are biomarkers that people care about using PPG, which is pretty much required and standard in most devices these days. But the last two are the interesting ones. Core body temp and hydration utilize our proprietary SWIR spectrometer on a chip. And that's in this wavelength range. Our next PIC expands the wavelength range to allow for additional biomarkers like alcohol, lactate, and glucose. I will offer in this meeting today that the general problem statement for product development, assuming that you're building something that the user needs and that the market wants, is that we need to reduce the time of this design, build, test, analyze cycle. Time equals money. So design, the entire weight of the product rests on the design phase. If you didn't have clear requirements, or if the design didn't meet those requirements, then the ensuing months of leading up to when you discover that in testing or during analysis is time mostly wasted. So to this end, there's a clear need for end-to-end modeling that helps translate clinical and system requirements to engineering requirements. On the PIC side, we have done quite a lot in design automation that can help predict wafer level yield. This, for a system, can be combined with optical models in commercially available tools like Zmax and model the light as it enters the tissue. And then the most important piece to all of us here in biophotonics is the tissue. We need a way to model light transport through tissue as part of this overall system. Now, all of these pieces are currently separate. There's lots of great, openly available tools, but they're not seamlessly integrated. And it's left right now to every person working in this field to try and put it together themselves. There have been some demonstrations of this capability of putting it together, like when I was at Triple Ring, but we need something commercially available to the industry as a whole in order to really mitigate the risk during the design phase. And I'll also posit that the maturity of this end-to-end model is really linked to one or more full development cycles. You have to get all the way through to the end to know if your model is in fact accurate. Moving on to the build phase, we'd like to be able to accommodate multiple build test loops for the inevitable surprises or miscalculations. And in order to do that, you need the build cycle to be as short as possible. And unfortunately for us in silicon photonics, wafer processing takes a really long time. It's great, of course, once you have your final design, you can make a ton of these things. But the process itself is lengthy. From tape out to an ASIC is 3 to 4 months. For the silicon photonic package, it's even longer. From tape out to a bonded PIC is 6 months, even with parallelization. And then it still needs to be integrated into the system. Broccoli has streamlined this flow as much as possible. So the IC line and the PIC line operate in parallel. Same with the silicon platform and then the 3-5 building blocks that go into it. The known good die is then integrated with the silicon platform at the end of the flow to maximize efficiency and yield. And the IC and PIC are packaged to achieve the final module for different functionality. The real challenge here, though, is that each one of these steps are handled at different fabs around the world. Epi growth, 3-5 processing, silicon processing, metallization, bonding, all different places. Managing the schedule conflicts and shipping through customs accounts for months of the time it takes. So an obvious ask for the photonics industry is a consolidated manufacturing flow. All under one roof would cut that time in half. And notably, this is how large-scale electronics manufacturing takes place, like think Foxconn or other vertically integrated contact manufacturers. So we need the same for photonics. And test, critically important. So there's myriad challenges in developing any one piece, but as most engineers know, anytime you develop 2 or more interdependent systems in parallel, no matter how good your requirements and ICD, your interface control docs are, something truly traumatic will crop up in integration testing. And so you want that to be as quick and efficient as possible. And there's also a lot to be said about scalability, going from 50 to 50,000 to 5 million. You need high yield and very fast testing, seconds, not minutes or hours. Broccoli has worked on high throughput visualization and wafer level testing and mapping. This is at the component level. At the system level, we have specialty built test stations, and here's where there's gaps industry-wide. For mature technologies like SPO2, there are manufacturer test solutions you can just buy. For new technology, the burden falls on how to test falls on the individual company. And what this means is that people who are working in the same or similar biomarkers end up testing very differently. And some time ago, in the diffuse optics academic community, there was a push for standardized phantoms and methods. That need is still present today across all technologies and biomarkers. Biophotonics community is unified through the examination of tissue, and importantly, we need better and more representative tissue phantoms. And finally, we come to the analysis phase. Effective analysis requires quality data, low reference error, and known endpoints. For new biomarkers, the question of whether or not we have the right answer is challenging when gold standards aren't available and we have to figure out how to process the data. Guidance documents often don't exist, which means clinical validation protocols don't exist and need to be determined. Those clinical studies are not quick or easy to schedule and carry out. They take several months at least. Algorithm development depends on that data collection. So you can see how one development cycle can take just so much longer than it should. And in this modern age, we also face decisions around calculating biomarkers on the device or in the cloud. If data travels over the internet, it's going lots of places, and to be useful, it needs to be combined with data from other sources. While developing rapidly over the last few years, this is still a new area, and we have to take extra care to ensure privacy and security and comply with all the latest and changing regulations. So this brings me to my wish list summary. Tissue Monte Carlo modeling that's built into commercial optical design tools, more design options to begin with, and these are very generic. I am sure that many other speakers today will talk about these things. Longer wavelength photodetectors with lower dark current, InGaAs, we got better options, broadband detector arrays, and better advanced detection schemes, architectures that allow for deeper penetration depths into tissue. As I mentioned, consolidate photonics manufacturing, have tissue phantoms that are more representative of physiology. That includes, layers, a blood network, flow dynamics, just really make it real and something that's standardized across the industry where possible. And faster route to clinical studies, weeks, not months, joining together on clinical studies, partnering up, saying, hey, I've got a wrist free, I've got a finger free, I've got an arm free. Let's tackle this very expensive clinical study with lots of people, with multiple companies all at once, and that'll be a time saver and cost saver for everybody. So with that, I invite questions.
Okay, thank you so much, Kate. This is actually very, 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 very cool. I mean, this gadget it could be like a big difference, especially because it's non-invasive. And I see a lot of your challenges over there. So one of them is going to be for the peaks. So I know we have a lot of people in the room that they do photonic integrated that they can help you. And also it's going to be a big challenge with all the databases and analysis. So it's going to be more computational. What else do you need? What exactly you are looking for?
Pardon, there's a chat. So I didn't understand your question versus the chats that are coming up. Can you repeat that?
Sorry.
Kate, the question is very simple. Can you identify for us, the photonics community, one or two challenges that could help you after this meeting? What are the first, number one and number two main headaches that you think some partnership could solve?
Well, I mean, at this stage of development, so I think that we've worked through many challenges to get here, many. And so at this stage of development, it's really about the latter ones that I've talked about, namely getting through, test and clinical validation, performing those clinical studies. I think when we're looking to next generation, that's where I'm really keen to have more available design options. Silicon photonics as a platform is just so capable. And it can, I mean, one of the reasons that I joined Rockley was because I've been in biophotonics for a long time, and pretty much everybody is just beating LEDs to death. Let's just face it. You just like, it's just an LED over and over again. And we have so many more things that can be done to make things smaller, more cost effective, and apply it to so many different application areas.
Mhm.
And so I think my personal interest in this is where can we go next, right? Like
I think for that we had the right person in the room, actually from FDA, from the agency that will actually help you bring the device to the market. Valentin Stanitsky is with us. Valentin, thank you very much for joining the meeting today. You have something in mind, correct?
Hi,
Valentin
everyone.
is in the room. Hello.
Yeah, can you hear me?
Loud and clear.
Well, I'm here to listen and to hear what photonics community needs. And I guess if someone is interested, they can follow up. But unfortunately, I cannot comment in the moment.
Of course,
I
but
will
we
give
would like
you-
to
I
let
will-
everyone know that you're here and you have a question that you told us in the chat. Could you tell your question openly?
Well, the first question I had is, we have President Biden has signed a new CHIPS Act, right? And it falls under semiconductor. Mostly people talk about semiconductors, but I don't know if photonics chips actually fall under this category. And are we looking at some nice boost in production in the US?
Kate, would you like to answer that? And I can give you an answer afterwards.
To my knowledge, I have not heard from anyone at my company that this will apply to photonics. It does seem very much geared towards the semiconductor industry. That said, it would be a very interesting angle to see what we can do there. And so I'll definitely follow up and see how that's written and if there is any ability to apply that to optics.
Valentin, Kate, we here from Optica have made a big study on the potential opportunities for photonics in both the European CHIPS Act as well as the CHIPS Act in the United States. And the photonics is explicitly mentioned in both CHIPS Acts. And today with NIST, they're already collecting expression of interest. If anyone in the room is interested on this, please contact me privately and I will give you the link so you can share your expression of interest on your need for photonics solution into semiconductor production. The next question in the room, Kate, you have a lot of questions, a lot of fans in the room. The next question in the room is coming all the way from Iris Light Technologies. Chad Husko, thank you very much for joining us. Could you tell us in one sentence what Iris does and then ask your question?
Yes, thanks, Jose. Iris Light is looking at a new way people are traditionally integrating 3-5 materials as active media. We are doing a photonic ink-based approach to making actives, that's detectors, LEDs, and lasers on silicon photonics chips.
Cool.
And so the question I asked is, like you said, you have some needs and those were pretty high level. And again, maybe this isn't the forum, but I'm just curious, like, what are some of the specs that you're looking for? And it looks like you're doing separate off-chip sources or bonded at least, separately manufactured.
They're all on chip. Everything's on chip. Yeah, so we actually bond the 3-5 directly into the silicon photonic chip, and we have our light sources and our detectors fully integrated. So we're actually just using the same material as detector. And I think that again for the very specific applications that we're pursuing right now, it is we get enough light back. We're able to get the signal. Where I'm looking towards is, more advanced, or not more advanced, but like something more than spectroscopy, right? Imagine you can do other things with the silicon photonics platform, in which case you would want to go towards off-chip detector solutions and things, imagine like imaging applications, things like that. And in the near and the SWIR bands, there's really not good broadband, small pixel count array detectors. There's a couple new offerings, and like Sony has one, and there's one or two others out there, but they're of course very costly, and they're still just very high dark noise. So I'm just sort of interested to know if there's other things that are being pursued, and again, what's the smallest size that we can make in that package.
Kate, stay tuned for many companies announcing big things at OFC. We have been discussing offline with many people actually from this meeting, but this is very highly stealth mode, so I don't want to give them the floor so they can comment, but stay tuned. There will be a company in this meeting that has something big to announce in that respect. Next question is coming all the way from Germany. It's coming all the way from Nanoscribe. They provide 2-photon polymerization. Jörg Smolensky, my friend Jörg is here with us. Jörg, what do you have in mind?
Hi, actually, it's not Jörg, it's his colleague Remy today.
Hey, Remy,
for
nice
him
to
right
see
now. Yeah. So my question is about tissue phantoms. Like, what are the challenges you're currently facing with tissue phantoms when it comes to the validation process, and what would you like to see in upcoming tissue phantoms?
Yeah, I can speak to that for a very long time. There are challenges. So ideally, you want the tissue phantom to be stable for a period of time. And a solid tissue phantom is not very, or it has difficulties in being applicable because you don't have a very good interface, right? You're putting two flat surfaces together, those aren't flat, and you're going to end up with an air gap that doesn't actually have the same index matching that you would against tissue. That's one problem. And then anything else that's sort of softer materials, with the exception of some silicone-based ones, are they don't last. You have to just keep recreating them. And then the other is that they're not. It's very difficult to actually create skin-like. There are layers, and there's blood flow in different layers, and some of that blood flow is pulsatile and some of it is not. In the wavelength ranges that we're using right now, we don't worry about melanin, thankfully, just because it's beyond that. But for shorter wavelengths in the red and near infrared, this is a major problem. And so you want to be able to represent melanin appropriately in the right layer. And certainly for being able to test the full system, that also includes, PPG, red and near infrared LEDs. That's an issue. So I'm really looking for something that is descriptive of skin that could be used by anybody who's looking at any penetration depth, any source detector separation, and really account for what's happening in pulsatile and non-pulsatile blood flow.
Here is the open challenge from Kate. If you can solve that challenge, please contact us and we'll make the introduction. The 400 people registered the meeting just heard it. The next question in the room is from a company that really means a lot to us. It's really one of these companies that provides near infrared time domain spectroscopy, the company Pioneers. We are really happy to have here in the room Michele Lacerenza. Michele, thank you very much for joining us. What do you have in mind?
Hi, Jose. Thank you for introducing me. I was wondering, since we are also working with devices in the medical field, how do you deal with modification you have to make to the device after clinical trials? So maybe you find an issue on the device during the trial, or you find out that you can improve the device in some ways to make it much better. And how do you deal with redesigning or retesting the device itself for FDA approval?
When you are going through testing, right, you have your DVT, your design verification testing, and then you move into, and typically as part of, or before you get to DVT, you're doing your EVT and you're doing your initial clinical studies to see if you meet those end requirements. Before you move to a validation study for FDA, you want to make sure in those run-up studies that you are meeting the requirements. Now, if you're not, and you discover, for example, that there was something wrong in your model, there was a tap ratio that was off for some reason, you just have to repeat that whole build test part of the cycle, right? That's why I showed that loop on one of the first slides there. And there's no way around. I mean, there's sometimes you can do things, and other times you just have to make a new chip, and that takes months and months and months. And so I think what's critical here is how can we have a better end-to-end model so that we can reduce the number of build cycles when you find those inevitable oops, and you have to go back, and it just adds months to your release.
Michele, now that you're
Yeah,
here, later on I will come back to you, Michele. If you could maybe show later in the meeting one slide about how you measure oxygen. I will be coming to you later because it would be great that people hear this from pioneers. Kate, the last question comes from the next speaker. We have Jakir Levin here with us as one of the end users, medical doctors. Thank you very much, Jakir, for being with us today. Tell us, what do you have in mind?
Yeah, thank you very much. And I was intrigued by your on-skin sensor technology. One of the challenges we have in my own clinical field of dermatology is excessive sweating or hyperhidrosis. And that can be a nuisance for some people, but it can be actually quite severe. People can't shake hands. They can't maintain a computer keyboard because it short circuits when they use it, and it can be life-altering. And people try all sorts of different treatments for this, and we do need to improve that as well. But one simple impediment to our studies is that we don't have a great way of measuring a quantitative sweat measurement in a reliable way. Sweat can be variable throughout the day, even in someone with hyperhidrosis. And some of our methods now, which include, a starch iodine test or simply measuring the amount of fluid or increase in mass of a piece of filter paper placed on the skin, don't provide a lot of quantitative information. And I'm wondering if your sensors might be able to help us with that.
That's an intriguing thought. I actually wasn't aware that that was a condition to worry about. I'll add that to my long list. So I will have to think hard on that one. So when you have a device that is in contact with tissue, right, as we talked about before, you have a physical connection, and assuming that there's any sort of moisture or any kind of fluid whatsoever, right, you're going to fully index match those two surfaces together, and you're going to fill that space. That space, however, is going to be really thin. So one of the things that we are measuring is hydration. We measure water quite well. But it turns out we have the opposite thing. We don't want to detect sweating. We're trying to measure the hydration of the underlying tissue. Mm-hmm. And it turns out the sweat is not really much of a problem because it's such a thin layer, and it kind of like wicks away, right? We don't gather it or anything like that. So it kind of maintains like just a constant interface. That's not a thing. So in your case, if you wanted to, you would have to have some kind of collection point so that you can quantify, an increase in path length over time or something like that. Yeah, I don't see a way around it. You have to collect the material. What was the challenge you said with absorbing pads?
Well, they're cumbersome, but also in reality, if you're doing, let's say, a before and after, you have some intervention that you're trying, and you measure someone's sweat before the intervention, and then after they've healed from the intervention, 2 months later or something like that, their actual sweating during those 5 or 10 minutes that you're measuring the filter pad is variable. There's an underlying variability to that. So ideally, we'd like to measure someone sweating over a longer period of time, like with a wearable sensor or something like that, and monitor that before intervention and then after.
Yeah, I don't immediately have an answer, but I'll definitely think on that.
Great, thanks.
Okay, so our one of our last questions is coming from is another of our speakers. So work for Diamond Tech, and actually is quite important question, and I think it's going to be also related with not only with the sensors but also the wavelength about the diff. So Werner, if you can go for it.
Yeah. So.
Yeah, can Werner? Yeah, can you ask yourself, please?
Okay. I have the general question to Kate. What is the typical penetration depth that you consider for your different sensing applications?
It is wavelength dependent.
Yeah.
You're working in the infrared. So what do you consider as typical penetration depth?
Well, typical depends on which band. We actually span a very large range with our lasers, very, very large. So in the shorter of our wavelength bands, it's millimeters, 1 to 2 millimeters, I would say. And then on the longer wavelengths, it would be shorter. It depends exactly on where you're measuring, but these are definitely again very standard. You calculate it based on tissue optical properties and the wavelength range. Yeah, and the source detector separation. Mm-hmm.
How do you distinguish different skin layers with very different molecular properties?
Well, it depends on what you're trying to measure. So when you're measuring, hydration, right, that's at the shorter wavelength range, and you're getting a much deeper penetration depth. And it's that particular span is not so wide that you're measuring something drastically different from one side to the other. And then when you're looking at the longer wavelengths, same with what you're doing, I imagine we'll hear more later, like for glucose monitoring. In that case, you're probing mostly, I would say, the interstitial layer between the dermis and the epidermis, as well as some of the upper dermis layer, is my best guess about where we'll be hitting.
Okay. Thank you.
So we move on to the next speaker. We go now to the medical doctor. We heard first the technology development. Now let's hear the real end user of what they need. We go to the Massachusetts General Hospital to meet Dr. Dermatologist Yakir Levin. Thank you very much for being with us. The floor and the attention of everyone, 400 people, is yours.
Thank you very much. It's really an honor to be here. Hopefully you can see my screen.
Crystal clear.
Okay, great. So again, my name is Yakir Levin. I'm affiliated with Harvard Medical School and the Massachusetts the Massachusetts General Hospital. I'd like to share a little bit about my background because I think that'll kind of help understand my perspective a little bit. It is a little bit unconventional for physicians in general, and particularly for dermatologists, in the sense that I am a clinical practitioner affiliated with MGH and HMS. And my subspecialty is not a particularly common one. I treat children with impactful birthmarks, as well as adults, using very high power laser sources or very high peak power. I also treat skin cancer and acne using very low power light sources. I also completed a PhD in electrical engineering as part of my MD PhD program. And my research program involves new technologies to try to develop new technologies to treat these and other skin conditions. But my interests extend, sorry, this is on auto advance, I apologize, my interests extend far beyond dermatology, in the sense that I collaborate with urologists, infectious disease doctors, gynecologists on development of devices to address problems in those fields as well. I also have an interest in the translation process in general. I participate in a program to educate clinicians here at MGH about the process of innovation and how to formulate their clinical problems in a way that can be meaningful for industry collaboration. And also work on enabling that translation. So I'll share just some thoughts about what matters to me in my own collaborations with industry, or what I would like to have. And they all center around the general theme of teamwork, but some specific things. One would be a genuine interest and commitment by the company leadership and by their research and development teams to improve my patients' lives. If the problem that I'm trying to solve isn't interesting to you and isn't something you care about or come to care about, then the chances are you're not going to really want to continue a collaboration and kind of really work through to a product that we can actually use. It's easy to kind of give up midway. I also like to have close involvement in product design and development. Not all clinicians will want this. Some of them will just some of them will just give you their problem, and you can go and solve it yourself and give them a product at the end. But I think that having this close involvement is both interesting to me and also can result in a better outcome. Another challenge that often happens is not between me and the company, but between the institution and the company. So the company has to be willing to work with the institution's intellectual property team and legal team in order to come to an agreement to enable our collaboration. And finally, we all have to have a combined strong understanding that an integrated approach is needed. It's not just me as a physician or even a physician scientist. It's not just a company with expertise in engineering and optics. It involves biologists and a whole team of people here at the hospital that are involved in doing these studies to validate technologies. One problem I'll share with you, but I'm happy to talk about others. Just an example. It's not specific to my field of dermatology, but it's related, is hospital-acquired infections. So you're probably aware that, people go into the hospital for a problem, but they can often develop in the hospital a new problem, an infection that they didn't start off with, but that came about because of something going on in the hospital. 1 in 31 hospitalized patients has that problem. Urinary tract infections are the most common, and they're because of catheters that are placed inside the urethra. So this is a non-gender specific drawing. So this is the urethra here, which kind of connects the outside of the body with the bladder, and a catheter is placed in the urethra and put into the bladder. Along with that comes bacteria. Bacteria crawl around from the skin, so that's relevant to my expertise, along the catheter into the bladder and can cause a urinary tract infection, which can proceed upward through the ureters, even all the way to the kidneys. There are 500,000 such infections, catheter-associated urinary tract infections in the US per year, with 13,000 deaths at a significant cost per infection. And because hospital-acquired infections are not reimbursed by insurance or by Medicare, it's up to the hospital to cover the costs of that. So the hospital has a vested interest in not having these kinds of infections. And the total cost in the US is more than $1 billion a year. There are limitations to current approaches. Of course, current approaches involve, being extra clean when you put the catheter in. There are some catheters that have antimicrobial surfaces, but whether it's because of expense or lack of efficacy, I'm not sure, but they have not been adopted. And I do think there's a significant potential role for photonics here in that antimicrobial light, in the 405 to maybe even all the way up to 470, preferably 405 to 415 nanometer range. By incorporating that wavelength of light into the device, we know that wavelength of light kills bacteria, and what we're really trying to do here is prevent motion of bacteria or prevent crawling of bacteria along a catheter. And in our lab, we've actually shown you don't need a lot of light for that. So there is the potential, I think, here for a very low cost, even disposable solution that incorporates blue light into addressing this problem. So I'm happy to talk about this or any other potential clinical issues. Thank you.
Thank you very much for a really great presentation. It is fantastic to hear the end user perspective and what are really your problems. I'm going to ask you, because of course, we're managing here a photonics industry network. So we'd like to translate your needs into photonics technologies when it comes to lasers, detectors. Do you have it's almost Christmas, right? Do you have a Santa Claus Christmas list?
Yeah, so I mean, the example I just gave is one example of integrating light sources, inexpensive light sources into devices. The one I gave of the urinary catheter is just one, but there are many other potential devices using different wavelengths of light where I would like a consumable, a disposable device with a very inexpensive light source with the ability to direct the light anywhere you want within the device. And that can be with a fiber, that can be with actual, the wall of the device could be a waveguide. There are many ways to do it, but bottom line is very low cost, low power, but integrated into a consumable device. So that's one thing. In the range of the laser treatments that I do on skin, let's say for children with birthmarks or for adults with cosmetic issues, we know the wavelengths of light, and we know the rough pulse durations and influences that we need in order to successfully treat these conditions. But it invariably requires a lot of trial and error in many treatments. And what we don't have is the ability to monitor during the treatment the impact of our treatment. We can see a difference sometimes before and after immediately, a change of color or something like that, and that is useful. But the ability to monitor temperature and blood flow, preferably at a several micron scale, but even if it's at a millimeter scale, would be very helpful to be integrated into our handpieces, into our laser handpieces. And then finally, I would say, and this, I don't know that this is specifically photonics, but the ability to automate these things, because once you do start getting high quality imaging with high resolution images, the ability to interpret those images can be challenging, certainly in real time. And so the ability, whether it's through image segmentation or just some other way with artificial intelligence of measuring the response and understanding what's happening at the blood vessel level, which is again microns, tens of microns, would be very helpful in an automated way.
Oh, okay, yeah. So exactly, what wavelengths are you looking for?
So a variety. In the I mentioned the blue light example was and in terms of what the laser wavelengths that we use for skin treatments, the relatively high-powered treatments, those range from visible light, usually in the green, yellow, red, to the near infrared, like 755, 810, 1064, and then further out, even all the way out to a CO2 laser, 10 plus microns. So we don't necessarily have to do any, you wouldn't necessarily have to build anything at 10 plus microns, but it would have to be able to be compatible with that laser in terms of measuring the responses to them.
Okay, so we start having here a lot of questions. So we have Gary Bishop. Can you introduce yourself? Tell us where you are from and your question, please. So it's related with the UV. Gary Bishop. Okay, so I will read it for him. So he's asking, like, have you tried to use deep UV around 250 nanometers? Oh, here you are. Okay.
Okay.
Thank you.
Hi, Akio. Just a thought coming out of the COVID piece that I work in the photonics industry for about 20 years. I'm a laser physicist by background. And we used deep UV at around 250 nanometers for sanitizing rooms and offices and stuff. And I wondered if you've thought about going that deep into the UV to sanitize the catheters before they're used. Obviously, there's an eyesight issue there because we're talking about a wavelength that basically does molecular bond breaking. So it's not nice. But would that help your process of, if the rooms are relatively sanitized, can you minimize that with the catheters by basically killing off the bacteria, ripping them apart, basically? Just as a thought.
So thank you very much for that. And I'll mention, you mentioned COVID. So I also built a robot using UVC to try to sanitize rooms during the COVID era, because one of our problems was having to wipe down rooms with chemical wipes all the time. So we built a robot using UVC to do that. But and if anyone wants to help us develop that, that would be great. But in terms of the specific, so I do think that those wavelengths can be helpful in sanitizing the devices before they're placed in the body. In truth, though, when we, these are sterilely packaged, they undergo sterilization before we even open the package. And yes, we have to be careful when we're putting it in the body, and it's possible that we are introducing some bacteria at that time. But for the most part, these bacteria actually enter the body while the catheter is already in the body. They're crawling along from the skin, along the catheter, and ultimately reaching the bladder. And then the issue of the mutagenic potential of that wavelength of light becomes an issue, I think.
Okay.
Yeah.
Thank you.
Yeah. Next we have Oren Gabe for OR light lasers. And it says it's the creation of this device. So, Oren, can you tell us how it can help, really can help, Jerry, for this? Hello? Is something wrong with the sound?
Oren, are you with us? Can you tell us a bit about the beautiful laser? We'll come back to you in a second. I think there's some problem with the sound. Helena will come back to Oren in a little bit, but this is something spectacular we want people to hear.
Here I am. I'm sorry.
Here he is.
Yeah,
Oren, what is a beautiful laser? Tell us.
Well, thank you for the opportunity. I'm talking from Buenos Aires, Argentina. I'm in South America right now. I'm a laser manufacturer since the last 20 years. I've been doing diode lasers for surgery, and lastly, I came up with this new invention, which is my patented device, which is a blue laser, a high fluence blue laser, 445 nanometers. It goes up to maybe 70 joules per square centimeter. And the first idea was to use this laser for superficial telangiectasias in the face and also on legs. We focus our the use of this laser basically for dermatologists and vascular surgeons. On legs, the situation is that we combine which is called sclerotherapy, the injections with the chemical agents, and then we use the laser to improve the treatment at all. On face, it works only with the laser. However, the use of the blue light, of the blue laser, has more and more applications, as here the doctor said, as a bactericide process with the blue laser, as well as an active acne, where we can reduce the inflammation drastically. We can see that after 1 week, you know, the laser helps a lot to reduce this inflammation of the acne pimps, I'm sorry. This is a new device. It's being commercialized in South America since the last year, and I have a US patent, and we are looking to start business in the United States as well soon. I will be visiting Photonics West in 2 weeks, so I will be glad to have to meet someone of you.
Thank you very much. Fantastic, Oren. Great to have you. And we can, of course, this is where we excel, trying to help our companies bring their products to the market.
Well, this is the device.
Hopefully, Oren,
actually, this is a small device.
Beautiful.
It's portable device, battery powered, right?
We look forward to seeing it further at Photonics West in a couple of weeks. Remember, the Optica organizes an executive reception there. Please contact us if you want to get in touch. Go back to nanoscribe, Jakir, because Remy has another question for you. Remy?
Hi. Yeah. So this is unrelated to antimicrobial because I actually don't think I can help you out with there. It's a bit of a too large size scale for us. But when it comes to other aspects of dermatology, what sort of challenges are you facing right now with minimally invasive techniques such as spectroscopy for trying to look at skin conditions, or when you actually have to do a biopsy, are you doing any innovation there to do better biopsies in order to go off for diagnostics?
So there are a lot of potential ways to go about, ways to improve those things. First, I'll mention one big challenge when it comes specifically to biopsies. So my background is not in optical imaging, but I did my PhD in electrical engineering in magnetic resonance imaging, pulse sequence development and image reconstruction and things like that. When it comes to skin, though, of course, when I first started, I was like, well, this is great. We can not so much with MRI, but other imaging technologies to address diagnostic and therapeutic questions in skin. But one challenge that always comes up is being asked, well, it's fairly easy to sample the skin to do a biopsy, and with not much cost, and if you can do that fairly easily, then number one, you need a technology. If you're going to use some non-invasive technology, it has to be as good as biopsy and also compete on price. And so far, that has been a challenge overall. That being said, I think there are potential applications here. For example, in the area of pediatrics, it is very difficult to do a biopsy, and yet children have lesions on their skin that are very, or we worry a lot about whether they could be precursors to skin cancer, and it's often hard to distinguish them. And we have to do a biopsy in order to determine whether something is, let's say, a Spitz nevus, which is a benign thing, versus a melanoma. And if we had a way to determine those things optically, that would be wonderful. I mentioned also in terms of imaging, our ability to monitor immediate responses to our laser treatments in a way that we can't currently. We just have patients come back every month, and we know that if we do it a certain way, then they'll improve over time. But if we can tailor if we can see the immediate response and even tailor our pulse parameters to the geometry of and size of the vessels that we're treating or the locations, then that would be helpful. And then finally, in terms of just regular adult skin cancers, right now, the best way to treat skin cancers is a technique called Mohs surgery. This is a technique where a surgeon removes a skin cancer and investigates the margins of the skin cancer intraoperatively, so they can take additional samples if necessary during the course of the operation and ensure clean margins at the end. I do think, it's a fairly cumbersome and almost prehistoric process, and that is a very expensive process here in the US, at least, and it's not even available in many countries. And being able to incorporate imaging to determine margins of skin cancer prior to surgery might be an area where even something that's relatively expensive could take hold.
Yeah, and do you consider too, when you're doing the biopsies, especially when you do large biopsies and you're exercising moles and et cetera, do you take into account the cosmetic effects afterwards? Because right now, or some doctors still only treat it as, you treat the thing and then you don't worry about cosmetics, but patients still have to live on with this for the rest of their lives. And sometimes they're actually very self-conscious about this.
Yeah, no, I do think, I'm sorry, I didn't understand your original question. Yes, that is a very important issue. So if we were able to get very much smaller samples of tissue, that is something that is an area of interest, both in terms of minimizing scars, whether it's a molecular readout rather than just a simple histological one like microscopy, could use a much smaller sample in order to obtain diagnostic information, and also recognizing heterogeneity of lesions. And when we take a biopsy, we're only sampling one area. And it may well be the case that there's important information outside of that area. And so being able to localize our biopsies better, or even being able to obtain information without doing a biopsy, would be very helpful. Absolutely.
Awesome. Thank you. And yeah, the last one was an add-on question. So you did understand my original question correctly. This just kind of came up as you were speaking. So thank you very much.
Thank you very much. Thank you, Nanosky, for your constant support of Optica. It is really great that you are so engaged with us. Jakir, this is a global meeting. We have people coming from Argentina. There is a company in Israel that joined Optica over the holidays. I want to welcome to the stage for the first time at an Optica meeting, Yaron Magal from ICAP, all the way to Israel. They scouted some potential cooperation with you, and they have one slide to share with us. Yaron, tell us how you can help the rest of the community.
Hello, good morning, good afternoon. You have my slide, or I have to bring it up?
You could bring it up, that would be great.
Yeah, I don't have a screen that I can't, you have to authorize me to do that. Oh, we can give it up if it doesn't work for you.
Let me see.
Now I got
my
it.
team get in touch with you.
Got it.
Yeah.
Got it. You have it. Well, great. Okay, just a minute. Yeah, okay, this is the slide. Okay, so good morning, afternoon, wherever you are on the planet, to everybody. Very pleased to be here. My name is Yaron Magal, and I'm heading the Israeli Center for Advanced Photonics. It's based on a research center that is usually, it's a government one with cherry picking of top scientists that are doing photonic components for the last 40 years. In the last 3 years, they also have the ability to manufacture the components themselves. We have a mini fab, if I would call it, which is very diverse. We do 3-5 semiconductors, either for lasers, all type of laser LEDs, and we can customize at will. We're doing specialty fibers if you're talking about passive fibers, or customizing fibers for laser fibers. Either it's in thulium that is becoming more important in the medical industry, on either in connectivity, connecting fibers and chips and all of that. And the idea behind it that we can have the whole spectrum of components for initial testing, and from risk reduction, checking new concept, and making small scale manufacturing, which is usually very important for medical application when they go through FDA and through the initial steps. And usually the big fabs are less keen to work with these high risk and low volumes opportunities at the beginning of the way. And of course, if it's grow bigger, we will help you to move it into a big fab. So this is the idea, to make all this vast knowledge accessible to the market. We have projects with leading companies. If you know Picodia, which is like Tyrannus, but doing it in a real way. This is a short way, but to explain it, they're really giving equipment to NIH and other already. So it's not only in their facility, it's working, and it's with a very, very, very cheap disposable. We're making for them a novel IP-based optical system to investigate the samples and diagnose it. Another example, during COVID, two of our researchers approached us with a new novel idea how to detect COVID in a non-culturing way. The signal is strong enough without culturing. I managed to get investment from the Minister of Health in Israel, and we demonstrated it's really working. And we have other medical application and other application. If you need prototyping of lasers, special customized fibers, we also do lab on chip. We have our own femto laser that we can make on a silica chip, both the fluid running and the optical sensor all in one chip. And like what Side Diagnostic is doing in Israel and all around the world. So we can customize it for you in all possible way. Just approach us. I'll be also in a Photonic Quest with some of my team. Very happy to be part of this community. And
we are
thanks
extremely
for
happy that DICAP was created, and we're extremely happy that we can now go to you for medium and low volume customization of integrated photonics, as well as ultra fast as writing on specialty fiber. Thank you very much, Jaron, for being with us. If many of you have been checking the news over the holidays, and I was, even though my wife wasn't too happy about that, you have seen that PacBio has really made a big announcement over the last years. For us, it is amazing to have with us one of the people in this industry that helped us for many, many years, Gloria Hoffler, VP of Engineering for PacBio. Thank you very much for being with us today. Tell us how the rest of the people in the room can help you be even greater than you already are. It is all about biosequencing. The floor is yours.
Thank you, Jose. Thank you for that kind introduction. So let me attempt to share my slides. And it's really an honor to be here with the rest of you. So let's see if this works, because let's see. Please.
Bottom of the Zoom window, green arrow up, and then you're an expert. The floor is yours.
Okay, did it work? Okay.
Perfectly.
Okay, great. Thank you. Okay, so first, maybe let me give you a little bit of background about myself. Gloria Hoffler. I've been in the photonics industry for, well, let's just say a little bit over 25 years, mostly in the optoelectronics and photonic integrated circuits. And I joined PacBio a year ago as VP of Engineering Research, and really to work at the intersection of biochemistry, hardware, and software engineering. So let me first tell you a little bit about who PacBio is, give you an overview about our technology, and then what we would like to hear from the industry. So PacBio develops and manufactures instruments and reagents for high throughput DNA sequencing. We're a publicly traded company. We've been publicly traded since 2010. And we have over, we have 1,000 instruments installed in over 40 countries with 8 global offices. Okay, let's see the next slide. Can you see the next slide?
Not yet. We still see the cover. Not yet. Can you press space bar?
Yeah, is that?
Yes, perfect.
Okay,
yes.
Thanks. Okay, great. So sorry, I can't. I joined late, so we've had some difficulties with the technology after all these years of COVID. But anyway, the backbone of our instruments is what we call the single molecule real-time sequencing, and that uses zero mode waveguides. So these are nanostructures that are etched on the surface of a semiconductor wafer. And then we use image sensors to simultaneously take movies of long single molecules of DNA. So that provides the most complete and accurate view of the genomes and transcriptomes and epigenomes. Our customers are pretty diverse, and you can see here in this slide, we work with human genomics all the way to plant and animal sciences. We work with large-scale genome projects, clinical researchers, epigenetics, complex, researchers that are working on complex disease, neuroscience, immunology, rare inherited diseases. Okay, I have to go with this. Okay, so over the past decades, we've had a portfolio of instruments that really speak to the technological innovation of being able to bring advances in hardware and semiconductors, as well as photonic integration. And really, it's been focused on two things. One is to increase the throughput so that DNA, the genome is quite large. It's 3 billion. Gigabases, and so we need to have increased throughput as well as increase the read length of the DNA. So we have hardware as well as chemistry innovations. So starting in 2011, our first instrument was able we were able to look at 75 to 150,000 zero mode waveguides, so DNAs on each one of these. And then with increased photonic integration, we were able to scale to 1 million. And just recently, as Jose had pointed out, we have now announced the release of our new product, which is Revio, which has 25 million zero mode waveguides per cell, 4 cells in parallel. So within 24 hours, we can get a run with 1 million ZMWs. So this is really transformational because it brings the human genome for long sequencing down to less than $1,000 per genome, per long genome. So this has really gotten a lot of interest from both academic and commercial labs. It really speaks to expanding clinical into the clinical area. Doing whole genome would become a routine for care of rare diseases and other genetic conditions. So we really look forward to impacting the industry with this product. So while most of what I will speak of today really focuses on the long read instruments that have a lot of photonic integration, we also have recently announced a high accuracy short sequence instrument that will also ship in 2023. So that's one of the reasons why we've been in the news lately. And please do check the website, and it'll tell you a lot more about all the advances on the instruments and what they can enable. But coming back to the long read sequencing, in the spring of 2022, the telomere to telomere consortium presented the first complete human genome. And prior to joining PacBio, I thought that the human genome was fully mapped, but it turns out that 8% of the human genome was left unresolved by the initial Human Genome Project in 2000 because of technological limitations. So long reads, in high accuracy long reads specifically, has finally removed this barrier, and and it's corrected the errors in the original human reference genome, and now has unlocked these complex regions for studies in variational and functional studies. So enabling, genomes, transcriptomes, and epigenomics in humans and other species. So this is really a big deal, and is one of the reasons why long-read sequencing, as part of the next generation of sequencing methods, won the Method of the Year for 2022. And you can see, of course, this is from various journals. So, coming to how do we do long read sequencing at PacBio? Our sequencing technology is based on direct real-time observation of DNA polymerase activity. So as you see on the first the pictures on the left side, we have the zero modes are these small wells or blind vias, as we would call them in the semiconductor industry. So they're smaller than the wavelength of interest. And so the illumination happens, and it's primarily at the bottom of the CMW. So here's where we capture the DNA polymerase, and we watch for that activity as the nucleotides that are tagged with dye are incorporated into the DNA. And so that then enables us with the CMOS image sensor to take movies as this incorporation is happening in real time. And that's what you see on the pointer. And that's what you see in these areas where you see the pulses that allow us to see whether it's an A, a G, a T, or C that is being incorporated. Not only do we watch are able to call the base, but we're also looking at how long it takes for the pulses, to come to incorporate. And this is what gives us direct information about methylation and epigenetics. And so this is really important because we get the information real time without doing any additional amplification. So that's part of the sequencing and the detection methodology. But one of the other things that PacBio has done really well is also to increase the accuracy of these long read sequencing, and we do that by applying what we call high fidelity sequencing, which significantly increases accuracy. And we do that by adding dumbbells to the original high quality DNA. So this circularizes the DNA, and then this DNA is sequenced, over and over in multiple passes to remove any random errors. And consensus then is built by the instrument using a hidden Markov model. So some of the benefits, from long reads is that the genome assembly complexity is less, variant detection, haplotype facing, which is important for reproductive medicine. So there's a lot of reasons why you want to really not just have long reads, but have very accurate long reads. Now, our history goes back to 25 years, and this is just a picture to tell you what what the demonstration looked like and how far we've come, right? So this is a lab at Cornell where our founders first demonstrated zero-mode waveguides and also the DNA polymerase template sequencing. So we've come a long way from that. As I show you first on the first instrument, which was the RS, it already had implementation of photonic integration. It was a fused silica substrate that integrated collimated optics with 0 MWs. And it allowed anywhere from 75 to 150,000 molecules to be detected. But the problem here, as I highlight with the color lasers, is that the optical train was really very large. It was meters long, so it had to be called around the system. So what you can see then is that photonic integration is really, really needed to to not just reduce cost, but also to to scale. Right, so our interest is in scaling and increasing the length read. So the first photonic integration products had cells that that require innovation in terms of processing in an 8-inch line with fused silica processing using 193 nanometer lithography. Then we moved to integrating directly. So this was using an off high definition detector camera. We then moved to incorporating image sensors. So we collect not just integrated the collimation and the optics, but also the detector. And so using that enabled us to increase the number of movies that we could get to 1 million. So 1 million CMWs at a time. Then from that, we continue to reduce and shrink the pitch between the pixels and the zero mode waveguides. And so decrease the node that we use on the semiconductor chips, and we moved to 8 million. And then as you can see, everything is integrated, the sensors, the rejection filters, the collimated optics. And then our newest system, it's a 25 million, 4 chips that can be done in parallel. And that also integrates a back illuminated image sensor. And then of course we have a number of other integrated sensors that are at different stages of development. So now coming to the opportunities, we have, in order to do this level of integration, we really work with a lot of the tools, the EPDA tools. And here is, we really leverage what's been done in the semiconductor industry and what has been done in silicon photonics at 1300 and 1500. And that's the opportunity as well. It has all been done at the wavelengths of interest for datacom and telecom. So shrinking from 10 microns to 3 micron pitch and even lower really required the use of all these tools, which today are not really interoperable. So we use ANSYS, numerical, and CMAX to design the waveguides, the grating couplers, the fiber to chip coupling, dipole emission, crosstalk, and the stack of the materials, as well as the thin film optimization software. We also use, Cadence and Mentor Graphics for the image sensors, and we put it all together. We're a small company, when it comes to photonics or to semiconductors, and so we use Lucida to do a lot of the layout and edit. We put it all together in a package that also leverages packaging, high volume packaging, but with very distinct requirements, because now we have in one single chip, a single consumable, we have all the interfaces to the instrument. So we have the wet chemistry, we have the electronics, we have the robotics to put the cells into the instrument and all the optics. So temperature and the types of blues that are used for visible wavelengths are sometimes not compatible with high volume assembly lines. So this is another area where we can use help. So two key areas, EPDA tools that have been optimized for the visible wavelength assembly. And then, if I may say also, I think something that that Rockley had also mentioned earlier, and that was, PDKs with foundries that are optimized for these wavelengths. So I think with that, I'll stop sharing. And
thank you, Gloria. This is amazing, because I mean it's not easy to get a long read sequence with accuracy, and this is going to be super helpful. So we have very clear that you have needs on integrated and also in design. So in your photonic integrated, what is your number one? What is you looking most in this area?
I think one of the areas that is of most use, it seems like my camera is frozen, but one of the areas of most use are really 3 areas: the interoperability and optimization of the tool at visible wavelengths from blue to red, the packaging. So today it would be also very useful to have integrated lasers, and that requires revs with the foundries, and all their PDKs are typically optimized again for 1300 or 1550. So PDKs in silicon photonics and silicon nitride, I should say, optimized for blue to red in the visible area. Those would be of interest.
Okay, so different wavelengths, different things. So I think we have someone from PyX here in the room, because I think they can help you. I mean, there is a lot of people here doing photonic integrated. If you start raising your hand and start
Erwin from Fix, all the way from the Netherlands. Erwin, it is great to see you. Goedemiddag. How can you help here? Do you have a challenge for packaging photonic integrated circuits for biomedical applications?
Yes, we have several customers in that space. I understand here it's really the challenge on integrating the laser with the core PIC. So we do hybrid integration. So I think there are certainly possibilities there to couple these two together. So right now you use an external laser, really like fiber coupled. Is that right?
That's correct.
Yeah.
That's correct.
Yeah, so there are ways to couple the laser source, the source, more directly to the PIC. That will simplify things. But it really boils down to looking more in detail at your design to see where exactly we can help. But that's certainly an area where we are very interested in to see how we can help you further with the integration.
Yeah, that's great. Yeah, as you see, the package is pretty small, so integrating directly onto the chip, it would be really useful as we try to scale up. But also it's difficult to do that within the constraints that we have in a cost-effective manner. So I look forward to solutions that you can offer there.
Yeah. What kind of volumes are you looking at? Is that something you can talk about? Or?
I think volumes in the order of 100,000 a year are this is not telecom or datacom.
No,
so that's probably about
Right, but it's certainly significant in the biomedical space.
It is significant. And one of the reasons is because we have consumables, and so these are part of the consumable part. And as I said, we have 1,000 instruments. And so if you look at that for 1,300 genomes per year, the consumables can add up pretty quickly.
Right. Is your cell, the picture that you just showed, is that a disposable device? So for every test that will be used just once?
It is a consumable. The use is not necessarily once, yes, though.
Yeah. So it's like, I see with multiple other companies in the space, this dilemma, are you going to wire it up with fiber or integrate the source, or do you find ways to remotely supply that and be that way cost effective, right? So yeah, that's an interesting dilemma to investigate. Yeah.
Also, when you think about firing directly onto the chip, you have to also take into account the environment. And these are biological labs. And while, in the telecom or datacom industry, we're used to, cleaning up the fibers and making sure that the connectors are okay. That's not something that's necessarily standardized in clinical or biological labs.
Yeah. That's why your choice to integrate the source as much as possible. And understood. Yeah, very interesting to talk more about that. To understand more about your specific needs. So
see if anybody is interested in collaborate. So we know now that Gloria has a lot of needs for photonics integrated, but also for design and proper simulations. So please contact us. We'll be happy, very happy to contact all of you. So Jose.
Is also really happy to contact all of you, and we are going now to Berlin to meet one of the companies that had made a difference on bringing quantum cascade laser technology to measurement of glucose to help the diabetics in the world. I am more than happy to welcome Diamond Tech to the room. Werner Mantele, thank you very much for being with us. The floor and the attention of all the community, from integrated photonics to quantum cascade lasers to infrared detectors, is here to help you succeed. The floor is yours.
Hi, my name is Werner Mantele. I'm the Chief Scientific Officer of Diamond Tech. Diamond Tech is a small company. We're about 20 to 25 people working in Berlin, with a few freelancers in addition to that. And Diamond Tech comes from diabetes monitoring technology, but we have developed a platform technology to monitor body fluids, processed fluids, fluids in fermenters, and so on. The technology we have combines mid-infrared quantum cascade lasers, QCLs. It combines that with a proprietary photothermal detection and specific techniques to get depth profiling as we want to look into skin, for example. And clearly we have developed clever algorithms to deduce the results from that. Now, can we, it doesn't move. I cannot get.
Try the space bar. Hit it harder.
Sorry, no. We got stuck here. Let me go back one, sorry. And try once more.
This is the problem of making the meeting live, because we are used to
now we have,
handle this kind of situation.
Can you
we
see
have
that?
companies like Quantium in the room. We have companies that really are looking forward to satisfy your needs on quantum cascade lasers, Werner.
Can you see that, or?
No, not yet. Can you share your screen again? Bottom of the Zoom window, green arrow up.
Once more.
And then select the window that you want to share with the world.
Okay, here we are. And now let's see if that moves. No,
yes,
we cannot, I cannot move to the second slide, sorry.
Okay, so we're going to share the slides from our side. Elise, do you have the slides of,
that's a good idea.
of Diamond Tech?
Yes, I have it in PDF.
Okay, here we go.
So I can.
No, he managed to solve it. Werner, continue, please go ahead.
Thank you.
The technology we have developed is very specific for glucose on molecular level. The basis is I'm an infrared spectroscopist. I've worked on infrared spectroscopy for close to 50 years by now. And I'm very much convinced, and we're convinced that the mid-infrared is a region where you have the highest specificity for glucose. So in the left side here, can you see my pointer?
Yes, we can see your pointer, okay. Yes.
This is a region around 10 micrometers wavelengths. In wave numbers, it's about 1250 to 950 wave numbers, where glucose has a very characteristic fingerprint. You see here spectra of glucose solutions in a range that is in concentration ranges that is very specific for diabetes patients, like 50 milligrams per deciliter to about 500 milligrams per deciliter. And you see individual peaks here in that range that arise from CO stretching and OH bending vibrations of the glucose molecule, extremely specific and easy to distinguish from other molecules that are present, for example, from albumin or lactate on the right side here. So we have a specificity by the selection of the wavelength. Now, the technology we have developed is a photothermal technology. Basically, you see the principle here on the left side. We use a quantum cascade laser to pump infrared light through this internal reflection element, IRE, into skin. This can be either a widely tunable external cavity QCL, or, as I show below, a QCL array that emits discrete IR wavelengths that can be selected. Now, once this infrared light penetrates into skin, and the penetration depth is about 80 to 100 micrometers, so deep enough to reach interstitial fluid, it leads to a vibrational excitation of the glucose molecules. And in the mid-infrared, vibration excitation leads to immediate de-excitation. That's about 10 to the minus 12 seconds. That's really fast. And it leads to a deposit of a small amount of heat in skin. And the small amount of heat in skin layers is proportional to the excitation power, and it's proportional to the glucose concentration. So as we control the excitation power, we have an access to the immediate concentration of the glucose molecules in those. And this heat developed in skin, deposited in skin, transfers to the internal reflection element. And in this internal reflection element, it creates a temporary thermal lens because we use pulsed excitation. And this thermal lens in the IRE in contact with the skin is about the same as you have at the air on a hot asphalt street in summertime. It leads to a mirage effect. So what we do here, we use a second laser, which is a simple red probe beam laser, and send it through this thermal lens. And it is deflected as soon as a certain amount of heat from the skin layers enters this internal reflection element. So the deflection of the probe beam is a measure for the concentration of glucose. And we detect this deflection here with a simple position sensitive photodiode. In addition, we use a depth profiling technique, which is a specialty of the photothermal technique, because as we use different pump frequencies, we can probe different layers. Briefly said, if you use a very low frequency for the modulation of the infrared pump beam, you can select the entire optical penetration depths, say around 0 to 100 micrometers. And if you use here a higher frequency, say, for example, 200 pulses per second, you probe selectively only the top layers of skin. This allows us to subtract the contribution of the stratum corneum, which is a layer that does not contain relevant amounts of glucose. So we have direct access to the glucose in the interstitial fluid of skin. Now, the technology works fine. We built a first model, a first device, which was complete in about 2019. It's shown here. It's a shoebox size multi-user device. So users can identify with an RFID chip here. The user gets his calibration curve, and this actually uses an external cavity tunable QCL, which scans the full glucose wavelength range and calculates from the photothermal signal, calculates the glucose concentration. The device is CE certified, and we have performed several clinical validations of that. I show you one of these clinical validations. We have performed an extensive study. We call that Study 100 because it included about 41 diabetes patients, type 1, type 2, and about 59 healthy volunteers in a wide age range, and about 50% male, 50 female. It was supervised by the pathologist, and we did for each of these volunteers, we did a complete glucose correlation or glucose tolerance test, meaning that we had about 2,400 non-invasive measurements, and we collected those with an invasive reference value. And all that was plotted here in the consensus error grid or Clark error grid. And you see that about 99% of the measurements are within the regions A and B, which means little or no deviation from clinical outcomes. And this level of accuracy, which corresponds to a mean error, is of about 11%. This is comparable to minimally or invasive standard devices here. So the principle works fine. The device is actually not made for sale, but we built this D-base model for clinical validation and clinical testing in a small series. Now, development has moved on. We have actually miniaturized the setup. We use here an array of quantum cascade lasers with a small number. Small means like 10 to 15 different wave numbers. In the mid-infrared, this is a device about the size of my fingernail here, and it allows you to get individual wavelengths that can be set on the peaks and on the minima of the glucose fingerprint. We also miniaturized the photothermal detection here. We have actually a measuring time now of about 10 seconds, and the device, the Deep Pocket, which is under development, the Deep Pocket allows about 50 or more measurements with one battery charge. It is actually intended to be the individual companion for the diabetes patient. It works, it's a standalone device. This means that the diabetes patient can use it as the only device for measuring, but it also can communicate with the diabetes app that we have developed. And it allows you via the diabetes app to even contact your diabetologist. So this device is under development, but I can present to you here the prototype. This is the prototype, 180 grams, about the size of a smartphone, a bit thicker here. And you see the sensor surface here, which is the internal reflection element. What you do here is you put your finger on the sensor, start the device, hold still for about 10 seconds, and after calculation, it takes about 5 to 10 seconds of calculation, you have the readout, which is your glucose concentration in the interstitial fluid, and that can be used by the patient to control the blood glucose very accurately. From here, the development moves on. First of all, the Pocket will receive a more advanced array of quantum cascade lasers with more lasers, higher power, which is due about to the end at the end of this year. And then we further miniaturize the device to go to a wrist watch device, which is needs some miniaturization, needs some integrated optics, but it's feasible, and we plan that for about 2025 or so. Let me briefly show you about the team here. This is just a part of our team. It's very complementary. We have people that are working in regulatory affairs. We have software developers. We have the electronics development, and we have strong partners. We have Samsung as a partner for the development here. We have NanoPlus as a partner for the development of quantum cascade laser arrays. And we have a global medtech player. I'm not allowed to give the name, but the global medtech player wants to apply that for diabetes patients and to couple that to an insulin pump. So that's about the end. I'm ready for your questions.
Can we just jump in, or?
Thank you, Werner. Yeah, no. Yeah, so thank you so much. Thank you so much, Werner. I mean, this is amazing. I mean, a lot of people, I mean, I have a lot of family that is diabetic, and they have all these things in their fingers, which is really bad, you know. Or you have, yeah.
So it's the pricking that's the problem. It's the pain. It's a small risk of infection. It's the costs, and people. I mean, the typical German diabetes patients pricks about 1.2 to 1.5 times per day, which is by far not enough to control your glucose value.
Yeah, and it's very nice. I mean, if it's non-invasive, that's really good. So we have Valentin from FDA asking again. So Valentin, if you are there, could you ask yourself your question?
Sure, thank you. So one of my questions is, so when it comes to different skin types, skin colors, skin textures, how does your device, have you looked at it, and what are your thinking in that direction?
The good thing about our technology is that the penetration depth is only about 80 to 100 microns, and the skin pigments are much deeper. So we don't have problems with pigmentation.
Smiling because we got this question after every Diamond Tech presentation. After every Diamond Tech presentation, we always get the first question. That was really great.
But you're right, we've tested that. Actually, the presently preferred measuring site is really on your finger because it's so easy. But actually, the finger has a stratum corneum thickness that is really variable, depending on whether you work with your hands or not. We actually continued a series of measurements, which is much better because here on the wrist, the stratum corneum thickness can be as thin as 30 microns. So we penetrate much deeper into the interstitial fluid, and the measurement is much more precise. So one of the possibilities is really to hold that on here, and it works better.
Valentin Stanitsky is here from FDA, and he's looking for challenges for the companies in the room towards meeting the FDA regulation requirements. Valentin, thank you so much for being with us. Werner, I'm going to take you to Rotterdam in the Netherlands, because we have here the Erasmus MC Medical Center, the hospital. Hamed Abbasi. Hello, Hamed. How are you doing? He has a question for you. Hamed. Well, I'm going to read the question because it's very simple. When will the device be ready for use by end users?
Okay, the deep pocket is about to be complete at the end of the year with a correct multi-wavelength laser. Then we do a series of clinical testing, which is probably end of this year, beginning next year. And we expect it to go to the market by Q2, Q3 in next year.
You know, Werner, words cannot describe how amazed I am by the progress of Diamond Tech. I knew you from the very, very beginning. I have goosebumps. The next question is coming from medical doctor and co-founder of Halitus, Sven Youngman. Sven, what do you have in mind?
Yeah, so thanks. I'm also massively impressed, also from Berlin, by the way, so we're neighbors. I'm wondering, so, you know, the whole trend with the continuous glucose measurement devices, that are patches, you put them on here, there's a small filament. Do you think you could compete with them when your watch is out in terms of like the lag time from measuring and doing continuous measurements and all of this?
The lag time of measuring through skin here is actually probably even shorter than the lag time of the sensor from the minimally invasive devices. So we actually had within our test 100, our study 100, we had quite a number of diabetes patients that had, in addition to fingerprinting as a reference, they also had this Freestyle Libre, for example, on their arm. So we had actually our non-invasive measurement with the D-base, the finger pricking as the official reference, and we had the Freestyle Libre running with it, and we could see that the lag times are either comparable or even smaller in that case.
We have many companies, Werner, that would like to help you. They are contacting us right now. When it comes to the cost of the quantum cascade laser, many people are asking us. We don't want to comment on that. Allow me to go to the next speaker, Werner, but once again, amazing. This is a success story of the European ecosystem. Thank you so much, Werner. And we continue. We continue to the field of OCT. We have with us Alan Wax from Lumedica, who's going to tell us about his new revolution in terms of lowering the cost while maintaining the high speed for OCT imaging. Adam, thank you very much for being with us. The floor and the attention of the potential OCT users is yours.
Well, thank you. And thank you, Jose and Tom, for inviting me to this session. It's been really great so far. Let me just share my screen here. Can you all see my screen? Yeah?
Gorilla Glass Clear.
Gorilla Glass Clear. I love it. So as you mentioned, my company, Lumedica, delivers low-cost OCT solutions. The work is based on research that came out of my lab at Duke University, where I'm a professor of biomedical engineering. We began developing this back in 2014 in stealth mode. Our first product came on the market in 2017. And I'm proud to say, you know, we're moving hundreds of units each year now in low-cost OCT. In case you're unaware, OCT stands for optical coherence tomography. And over the past 25 years, it's become the retinal imaging standard. On the left here, I'm showing a typical OCT image, where instead of looking at the retina en face, where you're looking at straight on, it's actually a cut through, where you're getting a tomographic image. You can see all the various layers of the retina. Before OCT, there was no chance to see this in vivo. But with the development of this technology and it becoming a routine diagnostic, you can now look for early signs of disease. And that's what I'm showing here on the right. Here you can see a diseased retina with a macular hole forming. Both of these images were acquired with our Lumedica low-cost OCT system. In general, we feel that the application of OCT has been greatly limited by cost. Typical devices may cost $50,000 or $60,000, and in the developing world, you might find lower ones that cost $35,000. But still, this is a significant cost and has limited access. But there are a lot of diseases that could be detected early and blindness prevented if we had access to low-cost screening OCT. And so I'm proud to say that Lumedica introduced the first low-cost OCT in 2017. Currently, we are selling the OQ LabScope 2.0, and it's available for $9,995. This is a complete OCT system. It's a base model with an engine and a scanner. It has a computer that's built in, so all you need is a screen, or you can even use your phone or a tablet. We have lots of options for any research applications you might need, whether you want to go up to 80 kilohertz, so faster images, better depth, better resolution, down to 2 micron axial, or even extended depth penetration at 1310. This year, we're going to be at Photonics West, booth 8529, where we're debuting the LabScope 3.0. And so we're increasing the A-scan rate to 34 kilohertz. It's an upgraded, improved instrument, and it'll be seen for the first time at Photonics West 2023. We have a lot of markets that we address with our low-cost OCT. A lot of them are in research areas, whether that's using it for developing new optical imaging techniques or examining industrial processes. It's really great for quality assurance. So if you've got a small assembly or device and you want to be able to image it, such as the tip of a fiber probe, OCT is fantastic for that. We sell a lot into OEM, where people take our devices and put them inside of their own system. So perhaps you've got a robotic surgery technique and you'd like to insert an OCT probe in there. We're glad to do OEM work. One of our largest segments is animal imaging. So animal researchers very often will measure large animal eyes or rodents to look at the efficacy of drugs or various treatments. Our device is uniquely positioned to help with animal imaging in that you really can't take a commercial OCT for retinal imaging and adapt it for animal imaging that well. In the clinical range, we're really focused on ophthalmology, although we have efforts in dentistry, dermatology, and endoscopy. All of these are ongoing. Our current device, the OQ iScope, is preparing for 510K clearance, and that we've done our clinical study, and we are writing up all the paperwork that's required by FDA. It's a beautiful instrument. I invite you to copy this link if you want to take a look at it in more detail online. We see this as a point of care OCT system where you can do screening very easily and cost effectively. It's very portable, and that this is the whole system. There's no hidden boxes under the table. There's no additional PC. A single person can pick this up and carry the scanner and the base unit under their arm, go from room to room, pack it in a case and take it with them. This is our proprietary OCT technology, so we've got several improvements in there which enable us to deliver such effective low-cost OCT, and I'll talk about those in just a second. This device is optimized for screening patients, so at the point of care, you want to have an initial screen as to whether or not you might have signs of retinal disease, and this system is designed for that. If you've got something more severe going on, then you might want to go see a retinal specialist where they might have a more full-featured OCT. But for screening everyday patients, we think our iScope is a great system for that. We did take home a Prism Award finalist for this a couple of years back. Didn't come home with a win yet, but still a beautiful machine. So diving in, our OCT capabilities, what would we like to see? We've engineered our whole OCT system from the ground up, and that's what lets us deliver it at such a low cost. So instead of picking the highest precision components for every single part in the system, we focused on how they all work together. So it was a system design. So ranging from the light source to the spectrometer to the optics that are used and the scanning optics, even the use of the computer and the software, they're all meant to work together. Initially, we did this off of, a commercially available, let's call them development kits or, packages that we could, put together. But at this point, we're making a lot of these components ourselves. We do all printed circuit boards, all of our software. Our real secret sauce, though, is the spectrometer. I believe we started off at the beginning with Kate Bechtel talking about agile design. We use agile design in our OCT systems. Back when I started in the field and you wanted to make a spectrometer, you had to machine all these parts out of precision, tolerance aluminum. Could be very expensive, very exacting. You need a PhD scientist to twist these knobs and put all the pieces in place. But I'm pleased to say that by taking a different approach, we're able to 3D print these spectrometers. And this is where our agile design comes in. If we want to make a change in this, we go into the computer, we type in a few changes. Let's say we want to change resolution or change spectral range. We can type it in, swap out a few parts, and print it again. This gives us a really unique capability for doing custom work in OCT. If there's a system design that you need for your application, we're able to make those changes really quickly. Our design cycle can be as little as 1 day. That is, if we see a spectrometer and we want to have a certain specification for it, we can make that change, print it, and be testing it that same afternoon. This gives us a really unique capability for meeting our customer needs. And what would I like to see from the field? We use silicon and InGaAs detector arrays in our spectrometers. There's only a few of them that are available for high speed, with the high speed that's needed for OCT. Usually in spectrometer, you're integrating for a long time, but the OCT spectrometers need to go fast. Our initial version was 13 kilohertz, but now you saw I have one for 34 kilohertz, all the way up to 80 kilohertz. If we had really fast OCT spectrometers with the unique design of a tall pixel, so typically 200 microns by 10, it would give us ability to innovate even further in the field. I'm showing a couple of examples here of sensors that are like this, but they're not really formatted for OCT yet. So, forming this into a camera, it would be great. The nice people at E2V deliver the OctoPlus, which is similar to this approach, but I'd like to see even more capabilities and speed available there, where these would go direct to USB, perhaps over a 3.0. Some of our parts that we use, again, our agile design process comes in really handy here. So you can see in front of our Lumedica OCT engine, we have the scanner. It's this 3D printed black handled device. Inside of it, we've got a couple of components that enable us to do rapid design changes. So we include a liquid lens, which lets us vary the focus during a measurement, focus in on an optimal measurement. And then we have large scan mirrors. So these are MEMS mirrors that enable scan ranges. Some of the things that I would like to see would be faster, larger mirrors. I know there's some basic physics there, might stop that, but doesn't mean I can't ask for it. And the liquid lenses, I think, if they were faster to respond, perhaps more robust to environmental changes, I think this would also be a useful device. I'd like to invite you to take a look at some of our OCT applications. If you wanted to jot down that number real quick, I'll go ahead and paste this in the chat if you want to check on it later. I'll finish up by saying if you'd like to learn more about Lumedica's low-cost OCT, you can ping me at adam.wax@lumedica.com, and I'm even posting my cell phone here. Thanks, and I'm open for questions.
Well, thank you so much, Adam. So I cannot believe that you can do that for less than 10K. Okay, that's really impressive, and I hope it really goes into the field soon, like going all around, because there are so many places that they don't have the access to have like big equipment. So it's going to be really, really great. So one of the things that I really need to know from you, so you say you need MEMS that are faster and obviously laser mirrors. What are exactly your needs? What do you need to get this real worldwide?
To be honest, what I need right now is FDA clearance for this device, but that's on me. I have to finish filling out the papers and send them over to FDA. One of the things that came up earlier was the need for optical phantoms, and you could really use a standardized optical phantom of the eye for validating scanning patterns, validating resolution. The FDA gives some guidance on this, but there's not really a ready commercial device, commercial phantom that would let us do these types of scans. It would be useful also for our research systems. I remember seeing that at NIST, they were working on OCT standards that would let you, validate resolution and depth penetration, but I'm unaware of those being available commercially. There are a couple of, phantom eyes that are available, but I wouldn't say that they were widespread available. And, their cost is also kind of prohibitive on wide scale use. So I'd like to see that.
Yeah, so from the photonics point of view, so what do you need to improve? What do you need to recreate? What do you need? Like, do you need, because at the end, I mean, once you get the FDA, it doesn't mean that you are going to have you are going to make it in bigger scale. So what is your step? What is your need for that?
Well, I feel our manufacturing process is pretty scalable. I mean, you know, the 3D printers are easy. You add another 3D printer, another assembler, and we're able to move those along. What I would like to see are more readily available high-speed line scan CMOS cameras. So things that go pretty fast, 40 kilohertz, 60, 80 kilohertz. And, you know, I think, am I still sharing my screen? Can you also, no, you can't see my screen. But, you know, the sensors
you can
that
share it if you want.
No, it's fine. You know, the sensors that I showed are generally available with, you know, I2C interfaces, and it's up to the company to develop an application for them. When the truth is, we'd all love to just have a USB 3.0 system come through there, and you know, the FPGA programming can be kind of difficult. So I would say more complete solutions for OCT spectrometers. I see a comment in the chat on 2D SPAD arrays. You know, I like those ideas, but I don't think they go fast enough. I mean, we're really pushing the envelope here on, you know, the speed. We at Lumedica generally stay within, you know, the 10 to 80 kilohertz range, but you know, we'd like to see them go even faster, 100 kilohertz, 150 kilohertz.
Thank you very much, Adam. In the beginning of the meeting, when we started, we had a quick question by pioneers, and I said that before the end of the meeting, they will show one slide. So before we go to Halitos, which is one of the highlights of the meeting today, I would like to give the floor to Michele Lacarenza again, so we can show in one slide the time domain near-infrared spectroscopy that you are providing at Pioneers.
Hi, everyone. I think I'm not able to share my screen. I'm pressing on share screen.
Okay, Elise, could you give him the right to share his screen?
And yeah, so as Jose was mentioning before, we are measuring tissue oxygenation and hemodynamics parameters in quite a different way. So we developed time domain nearest devices, and here is, if you can see it, is a picture of how the device is. It's a bit smaller than a shoebox. And what we do is injecting picosecond laser pulses in the near infrared region. So very short laser pulses, and we detect them by using quantum detectors, silicon photomultipliers mainly. And by looking at the changes in the shape of the pulse, we're able to detect the concentration of oxygenated and deoxygenated hemoglobin inside the tissue. And the depth penetration is different from what we've been seeing in the presentation right now, because we managed to go deeper, as deep as 2.5 centimeters. We range from 1 to 2.5 centimeters, and we have real-time information working in the domain of time. So we have a laser pulse, and the photons that are earlier in time tells us information. I think, do you still see me? Yes.
Yes, we do, and we hear you very well.
Okay. And so thanks to the fact that we are working in the domain of time, we have early photons bringing us information about shallow layers. So for example, the skin, in the case of measuring the brain, the extracerebral information, while late photons tells us about deeper brain regions. So we are able to detect the activation of the brain and how the brain is behaving by rejecting.
Thank you very much for this great presentation. Want to get in touch with this. We are really excited about the new startups that are joining Optica and developing this amazing product. So thank you very much. And now we go to one of the highlights of the meeting today. We always like to finish strong, and we're finishing really strong this time. This is one of the companies that got the most interest since I work at Optica, and it's been almost a year so far. Halitos, medical doctor Sven Youngman, and CTO Joanna Pavia, thank you very much for letting us go the way that we deserve with one of the highlights of the meeting. Halitos, the floor is yours.
Thank you so much, and thanks everybody. This has been a joy to watch. I'm here from Berlin. I'm a medical doctor originally, so don't expect me to go too much into the technical details. But I'm speaking to you from the user perspective on the end of the day, which is important for us in our development, that we closely intertwine software development, hardware development, and then the clinical perspective. I have with me Joanna, as Jose said, thank you for that, who, as you will see, is a fabulous engineer as well, and is mostly in charge of everything around the data perspective, but she also has a PhD in photonics. So she can take care of the additional questions. This is what we're building. I'll go into it in a moment. But the bigger story here is, we have taught machines how to see. We have taught machines how to hear. The next frontier in machine sensing, on our view, is smell. And there's a lot to be gained from that, right? The thing that we focus most on is the analytics of dynamic gases versus, your smoke detector or something. And within that, and that's also why I'm part of this whole journey, we want to look into the breath, to decode the breath. The larger big insight here that people have really been amazed by over the last maybe decade or even more is that every breath contains more than 1,000 molecules, 1,000 different molecules, I should say, right? And many of these molecules are volatile organic compounds, which reflect things that are happening in your bodies, your metabolism, and so on. And what's exciting about this is that it gives us a non-invasive, real-time, more or less, insight into a lot of things. You know that rats can smell diseases, or dogs and cats can. And so can machines. So a lot of the research that has been built on this and where we know these things from, they mostly come from the technologies around gas chromatography, mass spectrometry, and so on. And there are some other devices, you might have heard of e-noses, but the way we see it is they're all very limited when it comes to scalable clinical applications, because many of these devices are very sensitive. They need a lot of specialists to operate them. You have consumables that you need to use on end, and quite often they end up being very in sort of a centralized diagnostic setup, whereby sampling has to happen somewhere else. And by the way, sampling is one of the biggest concerns, because how do you know that what you've been measuring really comes from the breath? And how can you differentiate if it comes from the upper airway, the lower, or somewhere in between? Which does make a difference, because some of these biomarkers are produced in different parts of your lung. And what we've been doing is we developed essentially a photonics component and combined it with a gas dynamics component. So what you see here, the center, which basically looks like a Dyson blow hairdryer, if you will, this is a spirometry. So the thing you know from your lung doctor, nothing special. It detects the movements, the gases, and so on. And to that, we combine what we call the optical chambers, where this is just 6 meters in diameter, but it can reflect the laser up to 8 meters. So you have a wall of laser, essentially, and we put several lasers sequentially that are each triggered on very specific biomarkers, of which we know that we need to find them. So this is a tunable diode laser absorption spectroscopy. And in effect, what it allows us to do is we can know what the air looks like before somebody has been blowing into this. So we have a baseline assessment. And then we can do 10 cuts per second, which enables us to do a full end-to-end breath sequencing with data on the exact gas streams and with data on concentrations of certain biomarkers of interest. And the exciting thing about this is, and I can say that as a doctor, and we hate to say that quite often, is this will be able to be put in the hands of medical lay people. You don't need to have a medical degree. You don't need to be a nurse or a lab technician. You can be a security person at an airport. And the second cool thing is that we attempt to make this result show up in 1 minute. So if you compare that with, for example, a PCR for COVID, which in the express version, which is very expensive, is going to provide you with a result in 30 minutes, and somebody needs to swab you in the nose, and they need to send it somewhere. And we can say we do it in a minute. And we could even, send what we have here, like a geotagged signal onto a map, so that, authorities or aid organizations see in real time where the hotspots are in an outbreak. And this doesn't have to be COVID primarily, right? We can also talk about about tuberculosis, where still in Africa, 44% of the people with TB are not diagnosed and they keep infecting others. Or in the US, 20% of immigrants from certain regions have TB, right? So doing a quick screening there is quite interesting. Same for schools, you know, how do you make sure that children with infections don't come in and infect others and then infect their parents and so on, with a rapid screening, right? You can't do that with a 30-minute delay screening, or not even with a 15-minute so easily, not on a scalable basis. So that's it, right? This is photonics, we think is the way to go. It's the technology that is underestimated in the space. You know, people look at all sorts of chips, they look at mass spec and so on. But this is the only thing that really allows you to do an inline sampling, if you can even call it a sampling, because it's not really, that is easy to use, portable, and provides fast results. If you want to do a Q&A, so then...
No, we could continue. Joanna, take it over from here.
Yeah, so probably I will continue what I would think that it's a challenge or some challenge for us and also for the photonics community. So because as a background, as Sven presented, so I'm a biomedical engineer, but with a PhD in physics. So I've been working for the past 10 years in photonics technologies, but also with signal processing, machine learning, mostly for disease prediction and so treatment personalization, and also for predicting diseases in early stage, and doing personalized medicine. So there is a challenge that we are facing now, and I've faced so in the past. So because I was working also with optical fiber sensing, so trying to measure biomarkers in fluids like blood, using guided optics for diagnosing diseases like Alzheimer, for example. And now we are so I'm building this fabulous technology that is the creation of new digital biomarkers. So basically, you have, of course, the validated biomarkers like the proteins associated with these diseases. For example, in Alzheimer, you have the beta 42 for that is a protein specific that you can found in cerebrospinal fluid, and you have like optical fiber sensors to do that. So in this case, in breath analysis, you have volatile organic compounds, small molecule concentrations, and also lung function parameters. But the question is that there is a lot of studies and literature, so of course effective, but not effective enough in order to predict some diseases. So, and I believe, and I think that the scientific community believes that there is space for new biomarkers based on, for example, the derivation of these biomarkers. So, for example, signals related with the physics of particles, signals related with movement of for example, how people blow so the air into the device in the case of breath analysis. So the frequency, how they do that. So other physics related biomarkers that are not well validated yet. And so my question and my challenge is, at which level and when it's exactly enough so in terms of validation for the scientific community and also for the medical community, accept these new biomarkers as digital biomarkers also for using for screening in medical care and also for clinical usage. So
thank you very much, Joanna. And I can tell you that the reason why I'm so excited about this is because in my simple world, you are diagnosing lung cancer by simply measuring the breath exhaust with a handheld device. This is what you are doing. And this is so impressive. How can we help you? Because we really want to help you. I mean, I don't want to organize more telecom meetings at OFC. I want to help you. How can we help you?
I mean,
yeah, exactly. So please
no, you go.
Yeah, so this question about so this new digital biomarker scene, the explainability of the signals that we are getting, so of course that are based on science, but so they are new. So we need a new community based on with background on AI, but also on photonics, in order to help us to validate and convince the medical community that we have real information here. And secondly, so we are also building a big IoT infrastructure with centralized information, and we want that so this handheld and portable device, by, for example, 100 device could be able to deliver information to our cloud system and process and to do predictions. So for detecting lung cancer in 1 or 2 minutes. So this is a big...
Dr. Levin, when you see this, I mean, I don't know if anybody in the room is getting the goosebumps that I get when I hear this. What do you think the Massachusetts Medical Center could do to provide some help, some assistance of us having this device as soon as possible in our hands?
Well, that's a very good question. First of all, I think it is amazing and really wonderful to be at the point where we're going to detect these diseases, and especially lung cancer, with a simple point of care diagnostic. I think that's amazing. In terms of Harvard Medical School and Massachusetts General Hospital, certainly there would be great interest in implementation of such a technology. I don't know if you were asking specifically about, clinical trials or ways to bring it to the public. So we do have a clinical trials unit specialized in doing this. And I am not even though I do clinical studies at the research center and I do it on people, of course, that's related to skin, but I could certainly connect you with people if you were interested in exploring doing a clinical trial at MGH. Of course, you have many potential outstanding medical centers at which to do it, but yeah, congratulations to you, amazing, and I'm happy to help move things along.
One of the biggest consultants in our network is in the room. Richard Crocombe, consultant for spectroscopy solutions, is in the room. Richard, I don't know if you heard this and you get the goosebumps that I'm getting. This is a breath analyzer that can diagnose potentially lung cancer. How can we help? How do you think we can help?
A more general answer. I mean, I'm generally very excited by all the miniaturization of optical technologies and what that means for wearables, incorporation into consumer devices, all kinds of different areas. And I think that the things we talked about today in terms of silicon photonics, photonic integrated circuits, etc., have enormous promise in this area to bring the cost down, make things portable, make things accessible. And so I'm just
Richard,
generally very excited about the whole area.
Quantum cascade laser. So it is a quantum cascade laser. It's expensive. Yeah. It's expensive, but this guy needs cheaper quantum cascade lasers.
Oh, absolutely. I mean, historically, sort of one QCL chip was $10,000, from well-known suppliers in the area. And so bringing the cost of those down, and there are a number of people working on QCL arrays for various applications, including safety and security type applications. So QCL arrays, I think, the potential for low-cost QCL arrays, I think, are huge.
We also have in the room Shakiba Kabe from Oxford Instruments. Shakiba, what do you have in mind? So I'm going to read her question. Yes, Shakiba, tell us.
Yeah, I read the question, so I can answer. Yes, exactly. So Shakiba was asking me if we have to take in account the variability among the type of patients that we are including in our AI models that we are creating. For example, with our technology, we are creating models, for example, for detecting lung cancer, but for example, for the detecting and predicting exacerbation in COPD patients after they happening. So that is very valuable for physicians. So yes, we are considering cohorts with different parameters in terms of clinical comorbidities, so gender, sex, in order to test the models that we trained in an independent in a completely independent test and then we can prove that our algorithms could be robust in different cohorts all over the world.
It is about time now to close the meeting, to close the stream. Anybody who would like to stay, please feel free to stay, but we have been in YouTube for the last 2 hours. It is time now to close the stream and to close the public version of this meeting. So thank you very much, all of you, and especially thank you, Halitus, for closing the meeting in that spectacular